Welcome students. In this video we will study that mode of replication of DNA is semi-conservative. What is the meaning of this? Meaning is 50% of the parental DNA is retained in the daughter DNA or the new DNA. 50% parental DNA के ऊपर ही new DNA is synthesized, complementary strand is synthesized, and the experiment to prove that this mode of replication is there, semi-conservative, Meselson and Stahl, they performed the experiment. So let's study the experiment in details. They used heavy isotope of nitrogen for their experiment and E. coli. Meselson and Stahl's experiment. Scientists जो हैं वो they use radio isotopes for their experiments. In this experiment they used nitrogen. Normal is N14 and heavy is nitrogen 15. So heavy nitrogen was used in this. Normal nitrogen and heavy both were used in this experiment. They cultured E. coli, Escherichia coli, in a medium containing ammonium chloride, heavy ammonium chloride, for many generations, so that there are two strands of the DNA, so that both the strands produced by this E. coli, they have this nitrogen. Nitrogen is a very important component of DNA, compulsory component of DNA. So both these they ensured that are of N15 type for many generations, 14 generations. Now they extracted these E. coli from this medium and they transferred it into a medium containing normal nitrogen, ammonium chloride. So these they transferred it in a normal medium, and these bacteria they multiply in 20 minutes. Okay, so after 20 minutes they extracted this and measured their density. I'll tell you how. So this DNA must have produced the new DNA. So the new DNA must have used the new culture medium nitrogen. So this DNA has something from the new culture medium, means N14 must have been used. So this is N14, yani this DNA is hybrid DNA. It cannot be seen, it can be only measured. So how they did measure it, we'll see now. After 40 minutes they again observed the density of the DNA which is produced. Now you can see that these strands, since they are lying in the same medium, they must have used the nitrogen 14 type. So they will be hybrid. These are also hybrid. They are lying in the same medium, and 15 type of medium and 14 type of medium. So here the medium is same, so they will use that medium only. So these are N14 and N15 type both, and these are both are N14 type. Again this is N15 and this new is N14. Now measuring the density. So they used a cesium chloride equilibrium density gradient centrifugation method. Cesium chloride density meter mein they observed bands like this. These bands, when the culture medium was N15 type, and they are observed here. In hybrid they are observed at a different place, and in 14 they are observed when the culture medium is normal nitrogen, light nitrogen, the bands are observed elsewhere. So in the hybrid DNA, the density was intermediate, neither N14 type nor N15 type, in between density was there. And in this also, the results were as expected by the semi-conservative mode of replication, means hybrid DNA is there and N14 type of DNA bands were also observed. So this was the experiment conducted to prove that replication of DNA is semi-conservative. Subscribe and forward the video to the class 12th students. Thank you.